Pre-production, planning the video. What are the specific hazards? Physical, chemical, biological. Your group is focusing on physical, foreign objects in food, for example, glass, metal, plastic. Chemical, contamination from chemicals, for example, cleaning agents, pesticides. Biological, harmful microorganisms, for example, bacteria, viruses, parasites. B. What are the key safety practices to demonstrate for each hazard? Physical hazards, regular inspection of food and equipment for foreign objects using gloves. When handling food, proper food storage. Chemical hazards, proper labeling and storage of chemicals. Safe handling of cleaning products using appropriate amounts of chemicals to avoid contamination. Biological hazards, hand washing, using thermometers to check food temperatures, storing food at the right temperature, cleaning surfaces to prevent cross-contamination. C. What is your script outline? Cross-contamination happens when harmful substances like bacteria, viruses, or allergens move from one surface or food to another. This can occur in various ways, such as when raw meat juices spill onto vegetables, or when a cutting board used for raw poultry is then used for preparing ready-to-eat foods. For example, if you don't wash your hands after handling raw meat, you can transfer harmful bacteria to other foods, leading to foodborne illness. The consequences of cross-contamination can be severe, causing sickness, hospital visits, and in extreme cases, even death.
Now, let's look at some simple but effective ways to prevent cross-contamination in the food laboratory. Technique 1. Always wash your hands thoroughly before and after handling food, especially raw meat or poultry. Use warm water, soap, and scrub for at least 20 seconds. This is the most basic, yet most important, step in preventing the spread of harmful bacteria. Technique 2. Use separate cutting boards and utensils for different types of food. For example, use one cutting board for raw meat and another for fruits and vegetables. Color-coded equipment can help you easily identify which tools are for which tasks, reducing the risk of accidental cross-contamination. Technique 3. Properly store food items to prevent contact between raw and ready-to-eat foods. Always place raw meats on the bottom shelves of refrigerators to prevent their juices from dripping onto other foods. Make sure all foods are covered or sealed in containers to minimize the risk of contamination. Technique 4. Regularly clean and sanitize all surfaces, including countertops, cutting boards, and knives. Use food-safe sanitizers and be sure to clean them before switching between tasks like chopping vegetables or preparing raw meat. Regular cleaning reduces the chances of bacteria lingering on surfaces.
To summarize, cross-contamination is a serious hazard in food laboratories, but by following these simple prevention techniques, washing hands using separate equipment, storing food properly, and cleaning surfaces, you can help ensure the safety of the food you prepare. Always be vigilant and remember, safe food preparation is key to preventing illness and keeping everyone safe.